Hello students and welcome to the lecture of Microbiology Paper 1, Chapter 1 Golden Era of Microbiology. Today's lecture we shall study about the contributions of Paul Ehrlich. Paul Ehrlich was a German medical scientist. Paul Ehrlich was born on 14th of March 1854 in Sturlhen, Germany, which is now a part of Poland. Carl Wigret, who was a renowned bacteriologist of that time, was the cousin of Paul Ehrlich, and he played an important role as a mentor in his life. Ehrlich studied medicine in 3 different universities between 1872 to 1877. The universities that he studied in were Breslau, Strasbourg, and the Freiburg universities, and he obtained his degree in medicine from the Leipzig University in 1878. Ehrlich's doctoral dissertation dealt with theoretical and practical aspects of histological stains. He experimented with the aniline-derived dyes, which were very newly discovered in those days. Paul Ehrlich is known for his pioneering work in the field of hematology, where he identified the number and types of cells in the blood, immunology, where he gave the concept of the side chain theory, and chemotherapy, where he discovered a magic bullet or a medicine for the treatment of syphilis. For his work, he was awarded a Nobel Prize in 1908, which he jointly shared with Elie Metchnikoff, and it was awarded in the field of physiology or medicine. Ehrlich was offered an early position as the head physician at the prestigious charity hospital in Berlin. While working in Berlin, he developed a new staining technique to identify the tuberculosis bacillus. Ehrlich also differentiated the numerous types of blood cells of the body and thus laid a foundation for the field of hematology. He also discovered that methylene blue can be used in the treatment of nervous disorders. After studying the nervous disorders, he switched to the typhoid patients and found that a specific type of a reaction is seen in the urine of the person suffering from typhoid, and thus tested various different medicines in order to reduce or to remove the fever. He also made valuable suggestions for the treatment of eye diseases. Between 1879 to 1885, Paul Ehrlich published around 37 scientific contributions about his research. The last publication was of importance for Ehrlich because it dealt with the use of chemicals for the treatment of syphilis. At the age of 24, Ehrlich suffered from tuberculosis. After recovering from the infection, he worked in a small laboratory for a few years, after which he went to the Koch's Institute, where he worked on infectious diseases, and he concentrated on the problem of immunity. Robert Koch had given the postulates to describe how a particular bacteria is responsible for a particular disease in the humans and animals. But very little was known how these bacteria cause the disease, and even less was known about how the body fights against the infection or how the body's defense mechanism gets activated against the pathogen and how the effect is enhanced in response to the pathogen. So Ehrlich developed a hypothesis to explain the immunological phenomenon, and this was the side chain theory, which described how antibodies are formed and how they react with other substances. Right? And so, after studying the side chain theory or how antibodies are formed, Ehrlich proposed or suggested the serum therapy to be used against various different infections. Up to some extent, the serum therapy was useful, but many infections, particularly those caused by protozoa, failed to respond to the serum treatment. And because protozoal infections did not respond to the serum treatment, Ehrlich thought of developing certain medicines using certain unnatural substances in order to treat the infection. And this marked the beginning of the chemotherapy. Ehrlich started experimenting and identifying and synthesizing substances that are not found in nature, but these substances could kill the parasites without damaging the host organism. He began with the trypanosomes, a species of protozoa that he unsuccessfully attempted to control by means of coal tar dyes. Because he was working with dyes, he used the dyes in order to treat the trypanosomas, but without success. After which, he used compounds like arsenic and benzene for the treatment, along with the other compounds. But the other compounds that he used were very toxic and therefore could not be used on humans and animals. In spite of the number of difficulties encountered by Paul Ehrlich, he did not fail, but he turned his attention to the spirochete, that is Treponema pallidum, the causative agent of syphilis. In the olden days, mercury was used in the treatment of syphilis. Later, in the 17th century, Cinchona bark was used by the South Africans for the treatment of malaria because the bark contained quinine. The Brazilians used ipecac roots for the treatment of dysentery. So, plants were the source of chemotherapeutic agents in the olden days. The research of Paul Ehrlich started the new era of modern chemotherapy. Ehrlich's ambition was to find a magic bullet, a chemical that would kill the specific disease causing organism without harming the host cells. The search for the magic bullet started in the year 1891 with the studies of anti-malarial effect of methylene blue. He cured 2 patients with some success, but this therapy was not superior to the quinine therapy that was used worldwide in those days. His small success was a decision-making step in his scientific career in the field of modern chemotherapy. Ehrlich concluded that methylene blue had affinity for the parasite. Although it showed low toxicity to the patients, it was effective in killing the parasite. In 1905, Schaudin and Hoffman had identified the causative agent of syphilis, which is known as the Treponema pallidum. He wished to find a magic bullet that could be used to treat patients suffering from the syphilis infections. Ehrlich turned his attention to aminophenyl arsenic acid, commercially available as a toxil, which was synthesized in 1859 by the French biologist Pierre Jaques Antoine Béchamp, right, which had reduced the toxicity of the arsenic acid. And this atoxyl was used in those days, that is in 1905, in Africa for the treatment of sleeping sickness. But use of atoxyl had intolerable side effects in the patients, which led to complete blindness of the patients. So Ehrlich guided expert chemists at the Georg-Speyer-Haus Institute in Frankfurt to synthesize derivatives of atoxyl. Derivatives that were prepared by the chemists were screened for both antimicrobial activity as well as the undesirable side effects of these compounds on the animals. He systematically synthesized a number of chemical compounds, but the success was very limited. In 1909, Ehrlich had made the 606th derivative of arsenic, which he called as the Salvarsan. This preparation gave a very high activity in mice which were infected by the trypanosomes. And because high activity against the trypanosomes was observed, Ehrlich used the same compound against the chicken or fowl that were infected by the spirochete, that is Treponema pallidum. The chicken were rapidly cured after the use of the 606th derivative of arsenic, that is Salvarsan. So Ehrlich turned to his ultimate target, that is Treponema pallidum. In 1909 itself, he performed his first clinical trial on 50 patients with late-stage syphilis using Salvarsan was conducted, and it ended with an impressive positive outcome. So Salvarsan was effective against the bacterium, that is Treponema pallidum, which was responsible for the syphilis in human beings. As reported by Ehrlich, Salvarsan was a cure for syphilis, and thus the use of Salvarsan increased worldwide. And when the use increased, there were frequent reports of side effects caused because of the use of Salvarsan. People started humiliating and accusing Ehrlich for his derivative, that is Salvarsan. So Ehrlich suffered for a small period of time, but this did not break Ehrlich. He tried to study why are the side effects seen, and he found out very soon that the side effects of the drugs were due to the rapid degradation of the drug to the toxic products when it was dissolved in impure water without basification or without alkalization. To overcome this, Ehrlich directed the chemical modification of Salvarsan to produce a water-soluble analog that did not require basification but retained its high therapeutic activity. The need to find a water-soluble derivative that could be administered via the injection was the need of time. Finally, in 1914, compound number 914, that is neosalvarsan, was derived, which was marketed under the name of Neosalvarsan, that is a new version of Salvarsan. Neosalvarsan proved to be safe on humans and animals but active against the Treponema pallidum organism, and thus the goal to treat syphilis with a chemical was accomplished. Ehrlich's work was recognized with 10 honorary doctorates and the Prussian Great Gold Medal for Science in the year 1903. Ehrlich received the Nobel Prize in Physiology or Medicine in 1904, and in 1940 the Warner Brothers studio produced a feature film about Ehrlich titled Dr. Ehrlich's Magic Bullet because of the high impact of Neosalvarsan. The antibiotic revolution shined by arsenophenamine was followed by the discovery of Prontosil by Domag in 1932. Sulfonamides were the proof that Ehrlich was right, that is, the dyes could act as antibacterial agents. With the appearance of penicillin as a result of the work of Alexander Fleming, Howard Florey, and Ernest Boris-Chain, arsenals were gradually superseded, but they did not disappear completely. Today also, injectable arsenic derivative millarsoprol, marketed as Arsobal, remains in use for the treatment of sleeping sickness. Trained as a physician, Ehrlich can be considered on the basis of his research career as an organic chemist, a histologist, a hematologist, an immunologist, and a pharmacologist. Because of his contributions to the society, honors were given to Ehrlich. The greatest distinction bestowed on Ehrlich by the Persian state was the title Privy Councillor with the predicate of excellence. Ehrlich was presented with honorary doctorates by the University of Oxford, Chicago, and Athens. He was given an honorary citizenship by Frankfurt, where the institute he founded still bears his name. Ehrlich suffered from an heart attack in December 1940. Later, he suffered from a second attack in 1915 when he succumbed to the heart attack. The London Times acknowledged Ehrlich's achievement in the opening new doors into the unknown, saying, "The world is in his death." Thank you.